So there have been many efforts in the history of medicine to transform the circumstances surrounding certain diseases and those affected by them. Because they are underserved, under-resourced, lack public and clinical awareness, are controversial, and unfortunately, in some cases, they are also stigmatized. The purpose of this panel is to ask, what can the Lyme disease community learn from the history of other diseases that face similar challenges? Diseases like HIV and AIDS, Alzheimer's disease, and tuberculosis. What actions were taken by these communities that we can learn from in our efforts to reduce the burden of Lyme and other tick-borne diseases? This next panel will be moderated by Ben Nemser, Senior Program Officer of the Cohen Tick-Borne Disease Initiative, along with 3 panelists. Dr. Alan Greenberg, Professor and Chair of the Department of Epidemiology at the Milken Institute School of Public Health, George Washington University, Director, District of Columbia Center for AIDS Research, Dr. Sam Gandy, Mount Sinai Chair in Alzheimer's Research, and Director of the Center for Cognitive Health and NFL Neurological Care, Dr. Janice Binimore, Columbia University Assistant Professor of Global Health and Microbiological Sciences. This panel will be taking questions from the audience. Please feel free to text in your questions at any time during the panel discussion. Please help me in welcoming these panelists.
Excuse me. Thank you all for being here. My name is Ben Nemser. I work with the Stephen and Alexandra Cohen Foundation. First off, I wanted to thank all my panelists. Thank you guys all for being here. And secondly, just want to also send a nice thank you out to Savi and Shelley for organizing this entire meeting. So let's give them a round of applause, and all the the team from Mount Sinai and whoever helped out with lunch. That was delicious. Thank you. So first off, I wanted, before we kick it over to the panelists, I want to give a little bit of background around Lyme, so we're sort of all on the same page. Some of the stuff we've already discussed a little bit earlier, so I'll move through these things fairly quickly. First, as we know, there's a fairly high burden of Lyme disease, reaching about 300 cases per year. The tricky part is we also have about 10 to 20% that have consistent or persistent symptoms, oftentimes called chronic Lyme or post-treatment Lyme disease syndrome. Unless you talk to Crystal, she will definitely not appreciate me using that term, but it also sort of, but to Crystal's point, there may actually even be a larger amount, percentage. But having that 10 to 20% each year, there's been studies looking at, there's about almost 2 million people that have these persistent symptoms over time that are in the population. So it's a fairly high prevalence. Unfortunately, we don't have very good testing, right? A variety of diagnostic studies show that it can be as low as 46%, or even lower. That's actually an average. So it's almost like a coin flip in terms of the sensitivity of the tests. And there's been minimal innovation over that span in the last 25 years. Unfortunately, there's discordant guidelines, right? So you've got the IDSA that's preferred by insurers. ILADS is a secondary guideline, or so it's like an alternative guideline. And it breeds a lot of confusion with patients and practitioners. And so it created what we call the Lyme wars. There's a discussion around how to get treatment, and there's a lot of divisiveness in the medical community, and unfortunately the patients suffer. I think one of the key challenges, and you can even look on the CDC's websites, they'll basically say there's no treatment, no effective treatment for PTLDS. So patients are also oftentimes left in the lurch. If they don't have an option, they're really at the end of their rope. So they have to find some other means of getting treatment. And one reason why we don't know is we haven't done enough clinical trials. And I know Dr. Lewis was mentioning it earlier. We're really, for people with persistent symptoms, we're only looking at a few clinical trials that have even been done. There's 3 here that are constantly mentioned in the guidelines, but this is basically it over the last 20, 30 years around these persistent symptoms. And you'll see that the number of people enrolled, there's only about 220 people enrolled in those studies. So very few RCTs, and they really only looked at in monotherapy, right? So understanding from tuberculosis and some other diseases, only looking at one antibiotic or sequential antibiotics. And so you see down there's a few other diseases, right? And tuberculosis can have 2, 3, 4, 5 different antibiotics, same with leprosy. And oftentimes those treatment durations are more than 2 years, right? And so as we know in the Lyme community, we've only even tested up to maybe about 2 months, 2.5 months, right? So we always get that adage that we're having too much long-term treatment, but in fact, many other diseases have much longer durations on antibiotics. Not that antibiotics are the only option, but it's just one factor to look at. And I think one of the big issues that we all see in the field is what's really happening. We don't really know the pathogenesis of what's going on. There's a few different theories. One being that there's some sort of systemic dysfunction or damage that people with persistent symptoms are suffering from. There may be an autoimmune condition or response generated by Lyme. And then the third is persistence, right? So oftentimes people think that there may be persistent infection, hence additional rounds of antibiotics may be necessary, similar to something like tuberculosis. And unfortunately, then you have that in a larger context of other co-infections or other tick-borne diseases, right? And so, unfortunately, like I said, clinical trials are very low. If you look at some of the other disease areas, this is over, I think, a 3-year period. That's a little bit of an error, 2007 to 2010. You see hundreds of studies in HIV, influenza, hepatitis, malaria, over that course, and then Lyme is sort of down there. This is a very long list. We're way down at the bottom, but probably less than 10. So a small number of clinical trials, and unfortunately, that's or sort of either caused or a manifestation of low funding levels. So NIH about average is about 25 million, and that really hasn't changed over time, over decades. And comparing that to other diseases, if you look at the number of cases to how much NIH is funding, number of cases for, let's say, HIV is less than 50,000 in the US, and studies investment by NIH is about 2.8 million. So that's how you get that number of $56,000 per case, whereas Lyme is down at $85. So it's orders of magnitude, 500 times more money per case of the disease. So there is certainly a need. And then just a few last things on the discovery of other tick-borne diseases, right? We've talked a lot about the other tick-borne diseases. In the last 20 years, we've discovered 7 new tick-borne diseases, pathogens. And like I said, we haven't had any new treatments, no new diagnostics in that same period of time. So, or should I say FDA-approved treatments. So it's a really big concern. And from a clinical standpoint, you see now these multiple infections. One recent study showed that basically tick-borne disease does not follow this one microbe, one disease germ theory, where about 65% of tick-borne disease patients produce immune responses to multiple microbes, right? So, and then just so you guys enjoy your weekend out in Connecticut and in New York, there's a study that just came out that about 56% of ticks that were collected in Long Island, New York, and Connecticut tested positive for Lyme. So it's worse than a coin flip. So if you got a tick bite, you're most likely got bit by a tick with Lyme disease. And then 19% of them had multiple infections, right? So it's really a deep concern. And so as we were talking before, there's about 16 different infections that ticks carry. Now, not all in the same single tick. Usually it's, I think, about 6 in Ixodes scapularis. But what's interesting is that the number of clinical trials conducted on patients with a co-infection is 0. At least I cannot find 0. I cannot find any. I've asked a lot of people, even the CDC, if they actually have any. And this is, of course, because when a study, you're trying to eliminate any other possibilities, right? So those patients are always excluded. So we're not really studying anyone that has a co-infection. Now, this is only my own research. I may be wrong. My lovely wife in the background will tell you I'm occasionally wrong, once or twice a year. Please don't give her a microphone to double check that. So I encourage anyone, if you have a study, if you know of any studies, please send them along to us. And I'd even go as far, I haven't researched it enough, but on the diagnostics are the same thing, right? When you're studying the sensitivity of a diagnostic, my guess is that we're not putting in a lot of co-infections in that to study. They're actually looking at people with just Lyme. Again, haven't verified that, but I'm pretty sure. So on to the discussion. Really, I outlined a few areas where over the last 25 years, incidence is growing. We've had minimal change in government and even private sector funding. No new vaccines are on the market. The inaccurate testing, and then we've had minimal changes in the guidelines or any FDA-approved drugs. So I'd love to hear initial information from, or at least some possible discussion points with the panelists here around a variety of different points. So why don't I kick it over to my panelists without further ado. So why don't you introduce yourself and maybe tell a little bit, a couple minutes about the history of the diseases that you're sort of representing, or your sort of field of expertise, and maybe highlight a few parallels that you might see in sort of the history of your disease with maybe some of the challenges that we're facing. So why don't I give it over to Dr. Greenberg. Thanks.
Well, thank you very much, and thank you to the conference organizers for giving me this opportunity. I've learned more about Lyme disease this morning way more than I knew before, and it's been very enlightening. My name is Alan Greenberg. My wife is here, and I am from the Bronx, and I did my residency at St. Vincent's Hospital in Greenwich Village. I was here as a medical student between 1981 and 1985, which I know is before a lot of you were born. And, but those were the initial days of the HIV epidemic, which caused me to spend my life so far and continue in the pursuit of prevention and treatments for HIV/AIDS. I spent the first half of my career, 20 years, as a commissioned officer in the U.S. Public Health Service at the Centers for Disease Control, about half the time in sub-Saharan Africa and half the time working domestically on the epidemic here. And then I retired from career number one, and for the last 15 years, I've worked as the chair of the Department of Epidemiology at George Washington School of Public Health, and I run the District of Columbia Center for AIDS Research, which is a consortium of about 230 HIV investigators from 8 institutions in Washington, D.C. So my life, I sort of tracked the AIDS epidemic as we've gone through it. The initial days, we call it, as I'm sure you heard this morning, the pre-HAART era, or the pre-antiretroviral era. There was a time between really all through the 80s until AZT became available where there was really no antiretroviral treatment available. There were treatment for co-infections, but there was no treatment. And it was really as those of you who are old enough to remember, the band played on in those days. It was really a catastrophic time. All of us living in New York knew people in our families, friends in this audience. There was a lot of people sick and dying because, of course, it was a fatal disease. And then what happened was there was an enormous amount of activism, and I think the incredible engagement of the community in Lyme disease is something that very paralleled HIV. There was a lot of scientific activism and activism from what WHO calls civil society and community to put pressure on drug companies, on the government to increase funding for HIV/AIDS, and that was very successful. And I think it was described this morning in its sort of legendary ACT UP and really street protests and going down to the NIH, and it worked. And an enormous number of effort and resources went into developing antiretroviral therapy. And by 1993, the HAART, or highly active antiretroviral therapy, came out, and immediately when it was rolled out clinically in the United States, started reducing morbidity and mortality. But then we became aware of the AIDS epidemic in Africa and around the world, and once again, scientific and community activism kicked in, and the inequities between treatment availability in the United States and lack of availability in the many parts of the developing, non-industrialized world at that point was called, was very successful. There was a famous Durban AIDS conference where this was brought out, very high profile, and indeed because of that led to the PEPFAR program and the Global Fund for AIDS, TB, which we'll be talking about, in malaria, which put an enormous amount of U.S. government resources and industrialized country resources into trying to scale up antiretroviral therapy. Another major development is the use of antiretrovirals for pre-exposure prophylaxis. It's actually much like someone who goes to takes malaria pills to prevent them from getting malaria, or birth control pills to avoid pregnancy. Antiretrovirals are now used for pre-exposure prophylaxis, and we still don't have a cure. People who are on antiretrovirals, it's lifelong therapy, and people can live normal, healthy lives, and we still don't have a vaccine. But the combination of antiretrovirals for treatment and which can render someone non-infectious. We don't have ticks, and it's largely human-to-human transmission. The combination of antiretrovirals for treatment and prevention has made the possibility of us thinking about ending the HIV epidemic. And right now, both in my world and domestically in the United States, there's a new national effort, which I can talk about later, to really scale up rapid antiretroviral therapy and the use of PrEP in key populations to perhaps end the epidemic in the years ahead. So I'll stop there, but I'll come back to those points a little later on. So
Great.
Thank you.
Thank you so much. Dr. Gandy?
I'm Sam Gandy. I'm the director of the Center for Cognitive Health at Mount Sinai, and I've worked for the past 35 years or so on Alzheimer's disease. I think probably the similarity with Lyme in the Alzheimer's story maybe harkens back to the 70s, when there were nursing homes full of elderly people dying and being given diagnoses that made no sense, hardening of the arteries, that sort of thing. Even when I was in medical school, Alzheimer's disease was thought to be a rare disease of midlife. It was only in the 70s when Bob Terry and Bob Katzman, working at Einstein, recognized that the common forms of dementia that were killing the elderly were pathologically identical to that associated with the early onset form, which had been known for some time. And that was the moment in which the Alzheimer's epidemic was recognized, and now affects, it's now clear that about half of the over 85 population have Alzheimer's disease. It's almost endemic. The evolution of our understanding of Alzheimer's disease and the funding of Alzheimer's disease is sort of took a slightly different path, but I'll try and summarize that for you. So the degenerative diseases were difficult to distinguish even in the early 80s. And it was only over the past decade or two when molecular markers, both plasma markers, spinal fluid markers, and imaging markers have made it possible to distinguish Alzheimer's disease from tauopathies, tangle diseases, or other late life synucleinopathies, other late life degenerative diseases. Now, the major initial sort of bolus of funding for Alzheimer's disease that sort of got the field organized came about 30 years ago now, when the first Alzheimer's centers were developed, and there were about 25 of those developed, sponsored by NIH, developed nationwide. And each center had some mandatory components and had some research components. The mandatory components were a clinical core, and a pathological core, and an imaging and biomarker core. And so over the decades, in the past 3.5 decades, there accumulated clinico-pathological, clinical radiological, and clinical biomarker information to allow us to now be able to distinguish most of these diseases during life. And that's now sort of where the field stands in being able to make these diagnoses. Now, about 20 years ago, there was an investment in imaging centers and eventually in clinical trial centers. And those have evolved over the past 20 years or so. Now, I'd say much of the sort of the bumps in funding for Alzheimer's disease came because there's the Alzheimer's Association. Really, it's one of its major goals is advocating for Alzheimer's appropriation from Congress, and that was ultimately successful in sort of bumping the number of the congressional budget from about $400 million to about $2 billion, which is where it is now. That was championed both by the Alzheimer's Association and by former President Barack Obama, who put in a series of presidential bypass budgets that allowed the increase in funding for Alzheimer's centers. The cost of Alzheimer's disease is a bit higher than as estimated for Lyme. I would say every patient with Alzheimer's disease costs someone $1 million. The average duration of disease is a decade, and the average annual cost of custodial care, if you look across the country, is $50,000 to $100,000. So say $500,000 to $1 million per person, paid for by either private funds or by insurance. As this biomarker and genetic information has evolved, one of the first markers or the first molecules that was targeted is the beta amyloid molecule that forms clumps called plaques and in the Alzheimer brain and deposits around the blood vessels. For the past, I guess, 15, 20 years, that molecule has been the primary target because that's the defining lesion for Alzheimer's disease. Everyone with Alzheimer's disease, that's actually modified a bit lately, but most people with Alzheimer's disease have amyloid plaques, or at least the pathological definition requires the presence of amyloid plaques. I sort of dance around this because it's now been recognized that now that we can diagnose the disease more accurately with biomarkers, that about a third of patients who look to Alzheimer's experts as though they have Alzheimer's turn out to have no amyloid plaques. And we're just now trying to understand what that disease is. So we have a mystery disease there that we're trying to understand. It seems to progress more slowly than Alzheimer's disease, but at the time of presentation, when it was first described, experts could not distinguish plaque positive from plaque negative Alzheimer's disease. The plaque negative is called SNAP. That's the abbreviation for it, suspected non-amyloid pathology. And there's a great interest in understanding how that develops. But by focusing on the amyloid molecule, we've now been able to develop either immunotherapies or enzyme or small molecules that modify enzymes and completely purge the Alzheimer brain of all detectable amyloid. And unfortunately, the disease progresses just the same as though we'd never touched it. So if amyloid is a good target, then we must, then that means we'll have to begin administering amyloid lowering agents far earlier than we're doing now. These studies have begun in the 50s, but we have to go back to the 40s or 30s. And the problem there is that once you get to that age, there are people who have amyloid plaque, who have positive amyloid scans, who never develop Alzheimer's disease. So then you have the problem of offering an expensive, risky therapy to people who will never get the disease.
Yeah, it's tricky because all these things obviously cost money, and so the more detailed the study is, the more costly it is. And then, like you said, with the challenges around funding, I think is the key. We're going to come back to this. I think everyone is sort of waiting in bated breath just in general. It's very interesting information. But I wanted to just kick it over to Dr. Binimore to give a little bit of history around TB and talk maybe a little bit any parallels that you see.
Thank you, Ben. And I also want to thank the organizers for giving me the opportunity to speak about tuberculosis this afternoon. So my name is Janice Binimore. I'm the Executive Director for the Center for Sustainable Development at Columbia University. And I'm sorry, I have a little bit of a hoarse voice due to a cold that I'm getting over. I've been working in the field of tuberculosis for 20 years. I started off as a molecular biologist at the Pasteur Institute in Paris, working on developing new diagnostics and new vaccines for the disease. And then kind of transitioned when I joined Columbia into global health, which was to put into action the things that we learn from the laboratory. So develop programs that can actually help patients. And my focus was mostly on diagnostics because that's what I knew the most. Brief history of tuberculosis in less than 2 minutes. Well, it's a disease that we've been living with for over 2,000 years. Actually, there are mummies in Egypt that we know had tuberculosis. We found TB lesions in the mummies. And the Greeks used to call it phthisis, and then, throughout the centuries, it changed names. Some of you may know it as consumption. And for the longest time, people thought that it was a curse. You know, when you don't know what it is, then let's just call it a witchcraft or a curse. And it took late 1800 Robert Koch in Germany to actually isolate that it was due to bacteria. And he was able to say that it was Mycobacterium tuberculosis. We now know that there are 3 bacteria that cause TB, tuberculosis, bovis, and africanum. And so as soon as we knew that TB was caused by a bacteria, it opened up the possibility of finding treatments. But the first thing that happened out of my old institute in Paris was the vaccine. So 2 rather tenacious people decided that they wanted to come up with a milder version of tuberculosis. And so in a very interesting story, both on how they developed it and how they tested it on human, which I don't have time to, unfortunately, they developed BCG, Bacille Calmette-Guérin, which was shown to be effective as it protected children who were born of parents who had tuberculosis. And so throughout Europe, people started getting vaccinated. And the US did not follow suit, and that's a whole other interesting political story as to why not. In fact, all of you Americans are not, and all of us European-borns still are vaccinated for a very funny reason. Anyways, so in 1945, over in New Jersey at Rutgers University, there was the first antibiotic that was discovered called streptomycin, and that gave hope that finally we'll be able to eliminate tuberculosis. Had horrible side effects. Most people ended up becoming deaf. In fact, the first case had such severe tuberculosis that he was like, all right, let's try. He was cured of TB, but ended up becoming deaf. And then over the course of 20 years, pharmaceutical companies were interested in following suit, and so they developed many antibiotics. And so we went from monotherapy to multi-drug combinations, because we realized that the bacteria is a really interesting beast. If you target it with only one antibiotic, it immediately starts developing resistance, and then you're losing that one bullet that you have active. The last antibiotic, and this is something that I would have told you 10 years ago. The last antibiotic was 1963, called rifampicin, and it was also the strongest one. And then nothing. And then basically the world decided, you know what, tuberculosis is going to be a disease of the past. And pharmaceutical companies decided to move away from TB and focus on other diseases. But then HIV came along. And in the 80s, patients with HIV started having tuberculosis, because it is a disease that 1/3 of us have and will never express because our immune system is strong enough. But in the context of HIV, which leads to the complete breakdown of the immune system, tuberculosis comes back. So in the 80s, 90s, and in the early 2000s, we saw a significant increase in the cases of tuberculosis. The vaccine, by the way, works only in children, not in adults, because if it did, then we wouldn't have a problem anymore. And then the issue is that for about 120 years, the only diagnostic test that we had access to was the one that Robert Koch used in order to determine that the disease was due to bacteria in the first place in 1870 or 1890. So the field was desperate in the early 2000s, and I think this is probably one of the parallels that I can see with the Lyme community, is that tuberculosis felt completely left aside. HIV took the spotlight, even though HIV caused a recrudescence of tuberculosis. If you looked at the split between the 3 diseases, tuberculosis was always the ugly child. One good example is when in the year 2000, the Global Fund was created. So this is a fund that allows basically low-income countries to be able to have access to diagnostics and treatments that are funded by basically richer countries. The proportion of tuberculosis funding was only 16%. Compared to the other 2 diseases. So just a few numbers to put it back into context. Yesterday, actually, WHO released their latest report. We are now not at 1/3 of the world infected, 1/4, so that's good. And it means that less people are infected. 10 million people developed the disease last year, and 1.5 million died. So tuberculosis now has the sad title of being the disease due to a single pathogen that kills most people in the world, more than HIV and malaria combined. And despite that, we still get less funding than HIV and malaria. So some of the parallels that I see is that the TB community always looks up to HIV and malaria and tries to figure out how can we do it better. And I guess maybe later on we can discuss on what we have learned in the TB community that the Lyme community can try to replicate.
Great. So I'd like to ask a few questions of the panel, but please text in your questions. I'm going to ask each of them one question, and I'm going to go to the audience. So Dr. Greenberg, can you describe some of the major U.S. government programs that are in place to fight HIV epidemic, including how treatment guidelines are set?
Sure. Well, first of all, I think one of the parallels with the Lyme effort on a larger financial scale is the contribution of both foundations, which I've learned about two of them today, and the U.S. government to fighting the HIV epidemic. We have the Elizabeth Glaser Foundation, the Elton John Foundation, AMFAR, which Elizabeth Taylor was involved in on the private side, and then the U.S. government has a very vigorous response. Domestically, there are many U.S. government agencies that work on HIV, but obviously, like for Lyme disease, CDC is responsible for diagnostics and surveillance and prevention and policy. HRSA is responsible for care. Many of you are familiar with the large Ryan White program, which provides care to people who can't afford it, publicly funded care. And antiretrovirals are expensive. They're $10,000, $15,000 a year, and so there's insurance. U.S. government makes sure that anyone who's HIV infected has access to care. And then NIH is responsible for research. So we have funding on both sides. I think that the spectrum of work that NIH does, I think the budget is about $1 billion a year. Don't quote me on that, but I think NIH has for HIV research, is it? Yeah. That's about right. And through that fund...
About 3, but that's fine. At least for my research.
Yeah, through that funds a real national and international network of research. So there are clinical trials units. People have heard ACTG, which develops and tests new antiretroviral medicines, of which we now have dozens that are effective and can be combined in various ways. We work in the HIV Prevention Trials Network, where prophylactic regimens are tested to prevent HIV, and there's HIV Vaccine Trials Network, et cetera. So there are the networks, and then obviously they fund individual investigator grants. And then they fund what I'm most familiar with, which I think has some parallels to what I've heard the foundation, the Cone Foundation funds. NIH funds 19 Centers for AIDS Research around the United States, which are responsible for promoting HIV research, coordinating HIV research, and developing the next generation of researchers. So the two lessons learned that I think I'd like to share are, one, the importance of engaging community. Community was there from the very beginning, and we really work alongside our community. We have, for example, in my work, we have a very active community advisory board. The director actually sits in our office and and holds us accountable to make sure that the research we're funding and working on is going to be relevant to the communities we serve. And that's a real principle of HIV that we try to respect both domestically and internationally. I think it's really critical that those the communities you serve are at the table helping to make the decision, sometimes holding us accountable, regularly holding us accountable. They see the world through a lens of an infected and affected community, and are not shy about saying when we get a little off course and bringing us right on course. I can give examples of that. The second, which I think is very helpful, is the role the Centers for AIDS Research play in developing the next generation of researchers. Young people have fertile minds and come up with new ideas that they can cross wires that some of us in our 60s might not think of. And so the hallmark program of the CFAR is not really funding research, which is done through investigator-driven grants and R01s and the networks, but really funding the next generation of researchers, the junior professors, the young academicians, to make sure that when we get old and retire, there are new ideas coming up in the new generation. And I think those two hallmarks of engaging community, which you already have, but in a real way, academics working with government and community partners at the same table on a regular basis in a meaningful and transparent way. And two, a focus not only on the research you're doing, but grooming that next generation of researchers. Our biggest program is a pilot awards program. We've given away millions of dollars to. I think those are two very helpful.
Yeah, I think that's key. Bringing in emerging researchers and also having that link between the researchers and the community. I think it's key. One reason why we have this event here is to bring in researchers, community, and a lot of, I think, young scientists. So, Dr. Gandy, one question for you. I noticed recently Mount Sinai researchers had published a study that was linking Alzheimer's with some infectious diseases. Can you talk a little bit more about that, including how this sort of new theory was accepted by the wider scientific community?
So it's long been recognized that patients with Alzheimer's disease have higher titers of antibodies, that is, the levels of antibodies in their blood against HSV1, against the fever blister virus. But it has never been, finding that late in life doesn't really give you any sort of temporal sequence as to when that person might have been exposed. It could have been exposure could have happened anytime during life. So this recent paper that to which you're referring is derived from an entirely different and sort of surprising area. And here we've been doing RNA sequencing of brains of people with Alzheimer's disease, trying to understand how networks of molecules might move together so that we could better target therapies. And in looking at the transcriptome, that is the messenger RNA, we were surprised to find that turns out in several major brain banks in the country, the Alzheimer's disease, the brains of patients with Alzheimer's disease have an overrepresentation of genes from HHV6 and HHV7. That's not the herpes virus, not the simplex virus, but human herpes virus 6 and 7. And these are viral genes integrated into the genome. So one parallel that comes to my mind is of SSPE and measles, where measles virus genes are integrated into the brain genome during measles infections during childhood. They sit there dormant for years, and in some subjects or some patients, they become reactivated and cause a late life degeneration called SSPE. So that's the sort of scenario that's sort of a precedent for the kind of thing we're entertaining. I would not say by any means have we established that. We're really now trying to understand why it is all these brains have integrated these viral genomes. So the story is really very, very early. So part of the question was whether amyloid plaques could be induced by viruses. And actually, one of the things that are being done actively right now is sort of digging deep into amyloid plaques, TDP43 clumps from FTD, to see if viral genes can be found deep in those structures that might have been the inciting stimulus. So there's a lot of interest in it now, as I would say. It's still fairly speculative, and that basically what we found is a surprising number of viral genes integrated into the genomes of the brains of patients with Alzheimer's disease, and exactly why that is or when that happened is still a mystery.
Great. So Dr. Binimore, one question for you. You were mentioning around 2000. The tuberculosis community took a different strategy to support some diagnostic development. Can you tell us a little bit more about that and why it was important?
Okay, so actually really the shift happened in 1997 when the World Health Organization finally declared tuberculosis a global health emergency. So that opened up the way for possibility for new research and new funding coming into the field. Out of the three components, drug, vaccine, and diagnostic, it's actually the drug community that came together. It was at a meeting in Cape Town in 2000 that scholars and patients basically realized, okay, so it's been over 40 years that there have been no drugs. At the time, the multi-drug resistant tuberculosis epidemic started becoming more and more important, meaning that because of years of misuse of antibiotics, we started becoming resistant to, the bacteria started becoming resistant to the combination of drugs that we had. So we really needed new drugs. And so in 2000, the Global Alliance for TB Drug Development was created, and it's a public-private partnership, or call it a PDP, partnership for development of product, or product development partnership. And the idea was, in many pharmaceutical companies, or maybe in universities, there are compounds that are sitting on shelves, or in drawers, or in fridges, and that could have anti-TB properties, but are just not developed. The research and development costs so much, and a disease of the poor, like tuberculosis is, was not very interesting for pharmaceutical companies. So they were able to galvanize the field by re-injecting funding into the TB drug pipeline. And actually, thanks to them, we have now 2 new drugs on the market, one from Johnson & Johnson and one from Otsuka. And actually, about 3 weeks ago, FDA approved a new combination therapy called BPAL. So this is very exciting for the TB world because there is progress, and patients with multi-drug resistant tuberculosis now have hope that they can get cured. In 2003, looking at the TB Alliance, there's a group that got together in Geneva and created FIND, Foundation for Innovative New Diagnostics, basically following the same model. They received an initial 5-year grant, and the idea was also looking at technologies that may be interesting for tuberculosis diagnostic that a company was not interested in developing, again, because they didn't think that there was a return in investment. And so what FIND would do is they would do all of the clinical trial, they would do the tests in country and show sensitivity, specificity. And the contract was, if FIND takes your technology, you can sell it whatever you want in the United States, because there's a bit of TB in the United States, you can sell it in France. But if you do it in India or in sub-Saharan Africa, then it would be a subsidized price. And so that actually galvanized the diagnostic development and led in 2012 to the approval from WHO from the gene expert. You may have heard that if you are a little bit familiar with TB, which is the next generation of. So that was.
It's great. It sounds like a sort of like a public-private partnership. That
it is a public-private.
And then linking with sort of the advocacy. I think Dr. Spector actually kind of mentioned a little bit about that, sort of making sure that you're taking a little bit of ownership around that and pushing when sort of the private sector incentives.
And so the advantage is that it doesn't really matter who wins as long as there is one candidate that comes out of the pipeline. Mm-hmm. It's a win-win exactly. for the partnership. Yeah.
Savi's telling me that we're at time, but she can't get mad at me because I'm going to get one question from the screen that I saw before, which was around sort of the clinical research centers that I think you mentioned. If either you want to just talk very briefly about sort of what those are and how they're set up, sort of like a translational research center, sort of bringing sort of the research into the clinic and vice versa.
Right. So we're required to have cores, and we have an administrative core that sort of runs things and manages the funds. We have a developmental core that gives out pilot awards to junior faculty, which I talked about before.
By the way, we're coming from a context of Lyme, which doesn't have one of those. So how did it get started?
I think it started, I wasn't there when it started, but it was in the late 80s. I think NIAID started the CFAR program, and it's been successful, and it's been continued now for about 30 years. So we have an administrative core, developmental core, and we're required to have those. Then we have what we call our scientific cores. So we have our laboratory cores that provide basic science services to support researchers doing research, virology, immunology, proteomics, et cetera. Mm-hmm. We have a clinical core, which provide access to databases and specimens from patients who are willing to be in studies. And then we have a social and behavioral sciences core, which is social and behavioral scientists who provide expertise on methodology for prevention.
That's really interesting because you're basically bringing all of those communities together.
Correct. And
so that
we all
research and the clinical work happens.
Right. So before there was a CFAR in Washington, everyone worked in their own silos. They could collaborate with someone in Berlin or Australia just as well as someone down the hallway, but now
Yeah.
we all sort of know each other. And then the CFAR program, and I don't know about Alzheimer's, we have what are called scientific working groups where NIH asks us to constantly change every 2 or 3 years, but to pick focus areas where we put a lot of our resources to make sure we're not just, in addition to 1,000 flowers blooming, we're focused on specific issues. So for example, right now we have one group working on HIV cure research and one working on the End the Epidemic initiative. So we're trying to bring an end to the epidemic in DC.
Well, great. It sounds like that will be the last words. Just give a round of applause for our panelists. We really appreciate you coming down and sharing your list.
Can I just, one quick word of advice?
Yeah.
So the TB community was always lamenting why HIV, why malaria was getting more attention, and we quickly realized that we were just talking to each other, right? Every year we had the big TB meeting, and it was just TB people. So one of the advice that I would give to the Lyme community, go out, go to other conferences. I mean, so we are non-Lyme experts, but how many non-Lyme experts are in the room to try to learn from the disease? And how often does the Lyme community go out and do other conferences to try to basically be advocates to increase funding and to increase awareness?
That's a great idea, but my wife's going to be mad at you because then my travel schedule is going to go through the roof. But anyway, thank you guys so much.